Okay, we're in lab 17, the blood type lab. In this lab, we're supposed to do an experiment, but of course you won't do the experiment at home. So I will explain the process of the experiment, help you to understand how do we do the blood typing. So your job is read this menu, understand this experiment, and be able to answer these questions. Like how do we identify different blood type? We have blood type A, B, AB, and O type. And you should be able to answer these questions when you do the blood typing experiment. What do you expect to see? And I'm going to use this to explain the blood typing and the experiment. And you can also find these PowerPoint slides at the folder I put those lab menu. Okay, so we have the blood type A, B, O, and AB. And we also have the RH, but for explanation, let's use this. Let's forget the RH, because 90% of us have RH positive. And the blood type is based on the antigen. So antigen is the structure on the cell membrane. So if this red blood cell have nothing, that's blood type O. So if it's antigen A, it's blood type A. Antigen B, that's blood type B. If they have both, that's the AB. That's how we identify the blood type. And in this person's blood, you also have antibody. Antibody is in the plasma. So your blood, you have the blood cell and the plasma. So if this person have blood type O, in his plasma have anti-A, anti-B. And antibody will attack the antigen. Because this is blood type O, there's no antigen A, no antigen B. So he can have antibody AB in the plasma. So that's what in this person, blood type B person's blood, is red blood cell has nothing, no antigen, and his plasma have anti-A, anti-B. And if this person's blood type is type A, so he has antigen A on the red blood cell, in his plasma it won't have anti-A, because there's anti-A, anti-A gonna attack antigen A. So all the plasma he has anti-B. So if this your blood type A person, that's what you have in your blood. You have the antigen A blood cell and anti-B antibody in your plasma. And for blood type B person, they have the antigen B on the red blood cell and anti-A antibody in the plasma. And if this person is blood type AB, he has antigen A and antigen B, and he has no antibody, because if he has any antibody, anti-A, anti-B, you will attack it. So the blood type AB person, that's what his red blood cell look like, has A, has B, and no antibody. And we use the antibody and antigen match to identify the blood type. So this person, he is blood type A. He has anti-B antibody in this blood. If you put anti-A antibody, it's gonna bind with this and form a coagulation, and it will form the clot. And if this what really happened in your body, you're gonna block the blood vessel and damage your kidney. In some serious case, you can die. But that's how we identify the blood type. So if this person is type B blood and we put anti-B, we're gonna form the clotting. That's how we identify this blood type. So we use this antibody anti-antigen matching system to identify the blood type. So let's start the experiment. So in the experiment, we use the synthetic blood, and we have 3 different kind of unknown blood. And we have the antigen. That's the blood, red blood cell have antigen. We have the antibody, and we only have 3 kind of antibody. We have the anti-A, anti-B, and anti-RH, but this is a long word, so they call it anti-D, but actually it means anti-RH. So how do we test the blood? And we prepare the blood sample. First, we prepare the blood sample. So we put the blood unknown type A, unknown B blood, unknown blood number 3. Okay, we put those blood. Then what do? Of course, we add antibody, right? So we take the antibody, anti-A, anti-B, anti-C. And the same for this, the same for this. So we'll keep doing it. And what do we expect to see? If we add anti-A antibody, we see the coagulation. What does that mean? It means this person has A antigen. So by definition, he should be A. If you see the coagulation here, and means he has B antigen, and means by definition, he is blood type B. So if this both happens, this person's blood type is AB. That's how we identify. So if we see the clotting 1, 2, 3 positive, so we know this person's blood type is AB positive because the anti-D serum means RH. So that's how we identify it. So if we see 3 negative, it means there's no antigen A, no antigen B, no antigen RH. So this person's blood type is O negative. So that's how we identify this blood type. And that's what we really, exactly what we do when we identify the real blood type. So this is called the Eldon's car. So we take your blood and we just add those 3 different kind of serum. This because in the plasma we have the antibody. So it will bind with the blood. So like this one. So we see the clotting in A, we see the clotting in B, we see nothing in RH, no clotting. So that means this person's blood type is AB negative. And this one, we see the clotting in A, clotting in B, and clotting in RH. In the D is RH. So this person's blood type is AB positive. And if you see nothing, no clotting in A, no clotting in B, no clotting in RH, and this person's blood type is O negative. And if we see no clotting in A, no clotting in B, but in RH, this person's type is O negative. So that's exactly how we identify the blood type. Okay, that's it.